I'm Caroline de Theije. I'm from the University Medical Center Utrecht in the Netherlands.
I'm Gaurav and I'm from Gothenburg, and we are working with the PREMSTEM University of Gothenburg group.
I'm Claudia Verderio. I'm from the CNR Institute of Neuroscience in Milano, Italy.
My name is Henrik Hagberg, and I am from University of Gothenburg in Sweden.
My name is Mickaël Tanter. I'm a physicist. I'm coming from the Physics for Medicine Institute in Paris, working for INSERM.
We were trying to solve whether mesenchymal stem cells have an effect on the differentiation of neural stem cells on one hand, and on the other hand we were investigating whether mesenchymal stem cells effectively treat neonatal brain injury after inflammation and hypoxia or after intrauterine growth restriction.
So the question that we are trying to solve with the PREMSTEM is the role of mitochondria. What we are working on currently, that could the mitochondria transplantation in the brain possibly prevent the brain injury? That could be a very impactful work in the future.
The main question my research group tried to solve in the PREMSTEM was a basic research question related to the phenotype and function that microglia acquire when exposed to mesenchymal stem cells. And this because previous data indicated that mesenchymal stem cells can drive a protective phenotype in microglia, but no omic studies were available to understand which pathway were mostly influenced by mesenchymal stem cells. And this pathway can underlie transition of microglia towards protective function. Therefore, their identification is important to develop novel strategy to keep microglia in an homeostatic state and redirect microglia towards protective function during neurological diseases, including perinatal inflammation.
One of the problems in preterm infants is that sometimes they will get hemorrhages or bleedings in the brain. It's important to say that in the neonatal brain there is a particular region in the brain called the germinal matrix or the germinal zone. And this area have very fragile blood vessels. So during the acute neonatal intensive care, there is usually changes in blood pressure and in oxygen levels with hyperoxia, that is too much oxygen, or too low oxygen, what we call hypoxia. And all these risk factors together will, in combination with the fragile vessels, will sometimes cause bleedings or hemorrhages in the germinal matrix. So our question really was to try to mimic these bleedings in an animal model, in the rat model, to see whether these human mesenchymal stem cells would provide protection and reduce the consequences of these bleedings.
So in the PREMSTEM project, our goal was to invent an imaging modality based on ultrasonic waves in order to image the vascular system of the brain, first in preclinical models, in mice models, and then we would like to transfer it to human neonates.